Good morning. My name is Jonathan Hicks, and I want to welcome you to the NIH Office of Disease Prevention's Medicine Mind the Gap webinar series. This webinar series explores research, design, measurements, intervention, data analysis, and other methods of interest to prevention science. Our goal is to engage the prevention research community in thought-provoking discussions to promote the use of the best available methods in prevention research and to support the development of better methods. Before we begin, I have some housekeeping items. To submit questions during the webinar, there are two options. First, you may submit questions via WebEx by clicking on the question mark in the WebEx toolbar and direct questions to all panelists. Second, you may participate by Twitter and submit questions using the hashtag NIHMTG. At the conclusion of today's talk, we will open the floor to questions that have been submitted via WebEx and Twitter. Lastly, we would appreciate your feedback about today's webinar. Upon closing the webinar WebEx meeting, you will be directed to a website to complete a seminar evaluation and also sent an email with a link to the evaluation. Your insights will help us to continue to improve this webinar series. At this time, I'd like to introduce today's speaker, Dr. David M. Murray. After 35 years at the University of Minnesota, the University of Memphis, and the Ohio State University, Dr. Murray joined the NIH in September 2012 as the Associate Director for Prevention and Director of the Office of Disease Prevention. He is responsible for promoting and coordinating prevention research among and between NIH institutes and centers and other public and private entities. At this time, it's my pleasure to welcome Dr. David M. Murray.
Thank you. I'm going to talk with you today about a new resource that we have built called Research Methods Resources. It's related to the new requirements for clinical trials at NIH and particularly addresses trials that involve groups or clusters. I'll start by talking a little bit about the new requirements for clinical trials that NIH has rolled out. I'll speak a bit about group and cluster randomized trials, specifically and individually randomized group treatment trials. We'll walk through the changes in the application guide or application instructions related to these studies. We'll walk through the changes in the review criteria. And then finally, I'll give you a tour of the Research Methods Resources website, including an example or two of the sample size calculator that's available on that website. So NIH revised its definition of a clinical trial in 2014 after extensive public discussion. The definition is much broader than the definition that's often used in epidemiology and biostatistics, so investigators need to be aware of that. Any research study in which human subjects are prospectively assigned to one or more interventions, where the purpose of the study is to evaluate the effect of the intervention on health-related biomedical or behavioral outcomes, now qualifies as a clinical trial under this NIH definition. The four questions that you should consider as you're trying to decide whether your study meets this definition are listed here. Does your study involve human participants? Are they prospectively assigned to an intervention by the research team? Is the study designed to evaluate the effect of the intervention on the participants? And is the effect that will be evaluated a health-related biomedical or behavioral outcome? If the answer to all four questions is yes, then your study does qualify as a clinical trial under the NIH definition. There are new requirements for all clinical trials applications that are submitted on and after January 25th, 2018. You can find extensive information about the new requirements at the web address listed here. They include training in good clinical practice for PIs and project staff, the use of a single IRB if the application involves multiple sites. There are specific clinical trial funding opportunity announcements that you must use if you're going to submit a clinical trial application. You can no longer just submit an R01 under a parent FOA. It needs to come in response to a specific funding opportunity announcement that explicitly says it supports clinical trials. All clinical trial applications submitted on or after January 25th, 2018 will be evaluated based on clinical trial specific review criteria, and we'll talk a little bit about those. All studies submitted on or after January 2017 have to register in clinicaltrials.gov, and going forward, clinical trials also need to report results.
Registration should be complete within 21 days of the first enrolled participant, and results need to be reported within 1 year of completion of the study. There are new application instructions and forms, and we will talk a little bit about those.
I want to step away from the NIH requirements briefly and talk a little bit about different kinds of randomized trials, all of which can qualify as a clinical trial under the new NIH definition. The individually randomized clinical trial is the one that everyone is most familiar with. Individuals are randomized to study conditions. There's no interaction or connection among the participants after randomization. Many of the clinical trials that we do qualify in this category or fall into this category, but not all. At the other end of the spectrum from the individually randomized clinical trial is the group randomized trial. Now we're randomly assigning groups rather than individuals to study conditions, and we expect that the members of those groups have some connection before we ever show up to do the study. The connections continue during the study and even after the study. Examples are human trials involving randomization of communities or physicians or work sites, but these kinds of trials we also find in animal studies where perhaps we're randomly assigning litters of rats or mice or other groups to study conditions. In between is what we call individually randomized group treatment trials. Now we're starting off just like an individually randomized clinical trial with individual random assignment. However, the participants interact with one another or with a common change agent after randomization so that there is some connection among participants after randomization. So that is what separates this category from the first one. These trials are becoming increasingly common in public health and medicine. This graph shows the number of PubMed abstracts that identify group randomized trial terms either in the title or in the abstract. And you can see that it's gone from not very many in 1995 to lots and lots in 2015. It's almost an exponential curve. And these numbers undercount the true situation because many studies don't refer to group randomized or cluster randomized terms in their title and abstract. What's the impact on the design for using group randomization or for allowing participants to interact with one another after individual randomization? The individually randomized clinical trial, which usually has more than 100 participants, in that sense, there's a good opportunity for randomization to do its job and distribute potential confounders evenly among the conditions. That's also true in individually randomized group treatment trials, as many of those also have more than 100 participants. It's generally not true in a group randomized trial, where the modal number of groups in each arm is 20 or 25, and so the opportunity for randomization to work is more limited. The impact on the analysis is also important. Observations on randomized individuals who don't interact with one another or have any connection post-randomization, those observations can be assumed to be independent, and we can analyze them with standard methods that you've all studied in graduate school. However, the members of the same group in a group randomized trial will have connections, physical, geographic, social, something else, and they will exist before the trial gets started, while the trial is going on, and after the trial is over. The members of groups that we might create for an individually randomized group treatment trial will develop similar connections over the course of the study. They may not have any connection at the beginning, but we can see that interaction leads to those kinds of connections during the study. These connections create the expectation for positive interclass correlation, and that correlation violates the independence of errors assumption that underlies the usual analytic methods. As a result, we have to use special methods for these trials. When the interclass correlation is positive, the variance of the intervention effect is going to be larger than if there's no interclass correlation. The degrees of freedom available to estimate that variance are always based on the number of groups rather than the number of members, and may be limited. That's almost always true in a group randomized trial. It can be true in an individually randomized group treatment trial. If you ignore these issues of extra variation or limited degrees of freedom, you're going to have a type 1 error rate that's inflated, and often rather badly. As you can see here, a type 1 error rate of 50% is not uncommon in a group randomized trial, even with a small interclass correlation. And a type 1 error rate of 20 or 25% is not uncommon in an individually randomized group treatment trial. So these are circumstances that we want to avoid. Extra variation and limited degrees of freedom always reduce power, and so we need to take these factors into consideration as we're planning the size of the study at the very beginning. This is one of my favorite quotes from Jerry Cornfield. Randomization by cluster accompanied by an analysis appropriate to randomization by individual is an exercise in self-deception. So you've all been warned now, and I hope you will avoid that trap. His remarks were aimed at group randomized trials when he wrote that paper in 1978, but the general issues also apply to individually randomized group treatment trials. What is the state of the practice in group randomized trials and individually randomized group treatment trials? I wish I could tell you it was very good, but unfortunately it is not. We just completed a review of group randomized trials published 2011 to 15. Only about half documented appropriate methods for sample size. Only about half used exclusively appropriate methods for analysis. 30% used exclusively inappropriate methods for analysis, including methods that have been discounted and denigrated for 20 or 30 years. The situation is even worse with individually randomized group treatment trials, where reviews have shown that only perhaps 5% use appropriate methods. Poor sample size and analytic practices contribute to our reproducibility problem, and so the point of the resources that I'm going to show you today is to try to help investigators become aware of these issues and submit better applications. As part of the revision to procedures and policies that the Office of Extramural Research has done here recently, getting ready for applications that are going to come in on January 25th and later, we have revised the NIH application guide and the review criteria as they relate to clinical trials to alert investigators to the methodological issues inherent in group randomized or cluster randomized trials and in individually randomized group treatment trials. The clinical trial specific review criteria include similar changes. So let's walk through those. If you look at the new application guide, which is available on the NIH website, if you go to that website I showed you earlier on new requirements for clinical trials, you'll find your way there. Under approach, we have this language that's been added. For trials that randomize groups or deliver interventions to groups, describe how your methods for analysis and sample size are appropriate for your plans for participant assignment and intervention delivery. These methods can include a group or cluster randomized trial or an individually randomized group treatment trial. Additional information is available at the Research Methods Resources webpage. And if you're looking at this material online in the application guide on the NIH website, Research Methods Resources is a hyperlink, and it will take you to that website, and you'll get to explore the resources that are available there. In addition to this alert under approach, we have a similar alert under narrative study description. I won't read this one to you. You can read it on your own. We also have an alert under statistical design and power, again, very similar, alerting investigators that special methods are required if they're considering group randomization or delivery of interventions to groups. When we look at the new review criteria, we've added some language related to these designs here as well. Under study design, given the methods used to assign participants and deliver interventions, is the study design adequately powered to answer the research questions, test the proposed hypotheses, and provide interpretable results? So this is guidance that will be given to reviewers. We're going to be looking at your clinical trials applications submitted after January 25th, 2018. So you want to certainly pay attention to these issues since the reviewers are expected to pay attention to them. We also have similar language in the review criteria related to data management and statistical analysis. There are two general NIH-wide clinical trials protocol templates that are available. One is for phase 2 and 3 clinical trials that involve drugs or devices that FDA regulates. The other is for behavioral and social interventions. We are working with the people who are revising both of these protocols now to add similar language that I've just gone over from the application guide to the templates. So with that as background, let me now take you to the Research Methods Resources website and give you a quick tour of some of the material that's available there. As I said, NIH is launching a series of initiatives in 2017 and 18 to enhance the accountability and transparency of clinical research. And we provide links on this website to the clinicaltrials.gov registration and reporting policy, to the new requirements for submitting grants and contracts that are going to support clinical trials. We provide a link to the new forms and to the application guide. We provide a link to the new review criteria and to the single IRB policy. So all of these links would take you to the OER website where you can find out more information about the new requirements quite generally. As we continue on this particular site, we introduce you to material that's specific to trials that randomize groups or deliver interventions to groups. There's some background information, and then a subsection on group randomized or cluster randomized trials, and a subsection on individually randomized group treatment trials. You can learn a little bit more by reading this material or clicking on either link, and it will take you to material that's specific to group or cluster randomized trials or individually randomized group treatment trials. Here I clicked on the link for group or cluster randomized trials. You see the same figure that I just presented in my talk. There's also additional background on group or cluster randomized trials. And then we hit the resources that are provided on this website. There are a couple of webinars that relate to group or cluster randomized trials. One is a previous Medicine Mind the Gap webinar. The other is a 7-part online course on group randomized trials that's available from NIH. So you can use these links to get to those resources. We provide a link to the 2010 consort statement on cluster randomized trials, published in the British Medical Journal, so you can find that article. We have key references on group randomized trials, and they're presented beginning with the newest and working our way down to older papers. The last one on the list, one of my favorites, the paper by Jerry Cornfield, Randomization by Group, a Formal Analysis. But there are lots of good papers in between. There's a section that presents key references for state-of-the-practice reviews, so you can see how group randomized trials have been doing in terms of addressing the kinds of issues that I've raised. Unfortunately, you're going to see in general that only about half address these problems consistently, and that hasn't changed much in the 20 or 30 years that we've been monitoring this situation. There are also key references for sample size estimation for group randomized trials, many of which deal with very nuanced issues like what happens if, we have some very small groups and some very large groups. What happens if we have a dichotomous outcome as opposed to a continuous outcome? So many of these issues are dealt with in this set of papers, and I encourage you to take a look at them. We have a similar section for individually randomized group treatment trials. I'll just go there. As before, there's some general introductory material giving more background. There are links to the same 2 webinars because we talk about individually randomized group treatment trials in those webinars. There's a link to the consort statement that addresses studies that fall into this category. The title of the paper is Extending the Consort Statement to Randomized Trials of Non-Pharmacologic Treatment. Not all of that article deals with individually randomized group treatment trials, but some of it does. They don't actually use that label, so you may need to look a little bit, but you'll find it, for example, in Table 2, Item 7, as well as in the discussion of sample size on page 302. We provide key references for individually randomized group treatment trials, and any of these links will take you to the paper in PubMed, where you can read the abstract, and depending on your circumstances, you may be able to download a copy of the paper either for free or at some cost. We provide key references for state-of-the-practice reviews for this kind of design, and we provide key references for sample size estimation. As before, some of these papers get into nuances with sample size estimation. For example, what happens if people change groups over the course of the trial, or perhaps are even members of more than one group over the course of the trial. So I encourage you to take a look at those. We have a glossary on the website that defines many of the terms that I'm using and are used on the website. We have a list of all the references that are cited in this material on the website, and for most of them, there's a link to get to PubMed and see the abstract and potentially download the paper. We have a set of frequently asked questions. If you click on the little plus sign, you'll see the answer to the question, but there are quite a few questions here that often come up in the context of group or cluster randomized trials or individually randomized group treatment trials, and we hope that this information will be helpful to you. We have a feedback tab where you can give us your name and email address and then give us comments on this site. We're interested in knowing whether you find it helpful, whether you would suggest that we get rid of something or add something that we don't already have. Any suggestions that you want to make about the site, we would be happy to hear. The other item that's on the website is a sample size calculator for group randomized trials. We expect to add one later on individually randomized group treatment trials, but for now, the sample size calculator is for group randomized trials. We hope that this tool will be helpful to you as you're planning studies, or if you're looking at a proposed study and trying to get some sense of whether the sample size calculations were done correctly. You can use this page for either of those purposes. I'm going to demonstrate this sample size calculator with a couple of examples, and as you will see, there are 9 steps that we walk through for any sample size calculation. You can go back and forth among the steps without having to re-enter information unless you have explicitly overwritten something that you've already entered. The information will be retained even as you complete one exercise and want to go back and change something to do a what-if experiment and see what happens if you make that change. You can repeat the exercise as often as you want to compare different design and analytic plans or change parameter estimates. You can print the results from each exercise. You can also download the results as an Excel file or a Word file if you want to drop something from the exercise into a grant application. So with that as background, I'm going to launch the GRT sample size calculator, and but first I want to back up and talk about the example that we're going to use. So we're going to look at an example of what's the effect of a weight loss intervention on BMI. We want to implement the intervention in churches where we know that the people have been interacting with one another before the study came along, and we know that they will interact with one another during the study. So our unit of assignment here is going to be the church, and that way we don't have to be concerned about members of the church interacting with one another. We're not trying to keep them from talking to one another as we would need to do if we were randomly assigning individuals within a church to intervention or control. In this case, it's a group or cluster randomized trial, and we're going to, everybody in the same church gets the same intervention. The general idea going into this is that we'll measure BMI in 25 participants in each church at both pre-test and post-test. It may be that as we do the calculations, we'll determine that we need more or fewer participants, but that's a good starting point. We expect that the average BMI at pre-test among folks who are eligible for the intervention will be about 30. So we're specifically recruiting people who are overweight and obese. We expect the standard deviation to be about 6. We want to have 80% power with a 5% type 1 error rate to detect a reduction of about 5% from that average of 30, which would be about 1.5 BMI units. We know that BMI is highly correlated over time and expect that the correlation over 1 year would be about 0.7. And we plan to analyze the data with a mixed model analysis of covariance, adjusting for the baseline value of the BMI. As such, we expect to explain about half of the variance in the outcome by adjusting for pretest BMI. To be conservative, we're not going to assume any benefit for regression adjustment on the group component of variance, only on the member component. And now I will go back to the sample size calculator, and you can see here in the upper right-hand portion of my screen, here are the elements that we just talked about, and we're going to start entering those into the calculator. The first step is type 1 error rate and power. The defaults are 0.05 for type 1 error and 80% for power. That's what we said we were going to use, so I can simply press continue. The next step is setting the scale of measurement. Here, our outcome variable is BMI. That's a continuous outcome, so I'll click continuous. It asks me for the variance, and I said that the standard deviation was 6, so that the variance is 36. I can enter that. Then it asks me what analysis approach I might want to use, and you might not know exactly what I mean by analysis of a simple difference or analysis of a net difference, and that's one of the reasons that we've included these expansion panels on most of these pages. When I clicked on expand for more information, I'm getting a lot of background on analysis plans related to stratification, related to post-test only analyses or analyses that involve a pre-test and a post-test, and I get a description of what an analysis of a simple difference is versus an analysis of a net difference. In short, if I'm focused on the post-test data, I'm going to compare intervention and control at post, even if I'm adjusting for baseline, I'm analyzing a simple difference. It happens to be an adjusted difference, but it's still a simple difference. Only two means or rates or slopes that are involved. If I want to model change over time, then I'm interested in the net difference, where I'm modeling both the pretest and the post-test for all the participants, and that is something that we consider to be an analysis of net difference. In this case, I've decided to proceed with an analysis of a simple difference, so I'll check that circle and press continue. The fourth step is providing an estimate of the interclass correlation. This, if we expand the panel, this is the average correlation among participants from the same church in this case, from the same group more generally. But you can get an estimate from pilot data or from the published literature, and if you have pilot data, you can calculate it using the equation that's shown here. It's prudent to allow some level of interclass correlation in your calculation, and so if we close this box and look down, we allow a value of 0 if you want to enter that, but we think it's prudent to have some, and I suggested 0.05 for my example here, and so that's what I've entered. Enter the expected number of members or participants who will be measured in each group or cluster at each measurement occasion. That is step 5. Remember early on I said we're going to have 25 participants measured in each church, so I'll enter 25. We can change that at some point if I want. Do you plan to include regression adjustment for covariates? We said we would, and that pops open several other panels. The first of which is enter the degrees of freedom that are going to be used by any group level covariates. That's important because if I use group level covariates, degrees of freedom associated with those are taken away from the degrees of freedom for my intervention effect test, and I want to minimize that number. In this case, I'm choosing not to use any group level covariates, which means 0, and certainly that is the minimum. In terms of the proportion of the member level component of variance that in my outcome that I expect to explain, remember that the correlation over time for BMI is 0.7 at about 1 year. I'm going to adjust for baseline so that the proportion of variance is 49%, and I would enter that as 0.49. I'm not going to put in a correlation for any reduction in the group level component of variance because I don't have an estimate, and a conservative value then would be 0. Now I'm asked, do I want to consider the advantages and disadvantages of a priori matching stratification or post-hack stratification in the design? If I say yes, I'm given lots more options and material, but for this basic example, I'm just going to say no for now. So at this point, when I get to step 8, I have selected an unmatched, unstratified analysis. So the sample size calculations aren't going to reflect matching or stratification. And I'm asked to enter the hypothesized intervention effect, or delta, as a simple difference between two means, proportions, rates, slopes, or some other statistic. Now, I said that my intervention effect was going to be 5% from the baseline average of 30. So that gives me 1.5 BMI units. So I simply enter 1.5. And now I'm ready to calculate the results. I press the Calculate Results button, and almost instantly, I get to see the both a summary of the parameter estimates that I provided and the results that I've obtained. So I'm reminded that my type 1 error rate is 5% and power is 80%. I'm reminded that the variance is 36 for BMI. I'm reminded that this is based on an analysis of a simple difference with an ICC of 0.05, 25 observations included in the analysis in each group or cluster at the end of the study. I'm using regression adjustment for covariates, but not any group level covariates. And the proportion of variance that I'm explaining at the member or individual level is 49%. Nothing at the group level. I'm not using matching or stratification, and my intervention effect estimate was 1.5. So that's just a summary of everything that I've entered. Then I get the results. It turns out that under these conditions, I need 19 churches in each study arm or study condition in order to have 80% power for the effect that I've specified. I also get a little two small tables at the bottom. I'll try to scroll so that you can see both of them. The first one presents the absolute detectable difference, so this is in BMI units. The number of groups or clusters per condition is shown at the top, and the number of members per group is shown along the left. The cell that I proposed, which was I wanted to find an effect of 1.5 BMI units with 25 people in each condition, generates the solution of 19 as required, and that's in the center of the table. The point of the table is that you can see what happens if I add a few more groups or if I lose a few groups. What happens if I add a few more members or lose a few members. And so what you're getting is a detectable difference estimate in BMI units for all of these combinations. So if I increase to 20 or 21 groups and still had 25 members per group, the detectable difference gets smaller, 1.4. If I lose a few groups, the detectable difference gets larger, and that's a general pattern. We also show in the table at the bottom the results in terms of standardized units. So these are standard deviation units. The 1.4766 BMI units translates into 0.2461 standard deviation units. Some people are more comfortable with standard deviation units, so we show both. Scrolling back up a little bit, it's certainly possible for you to print these results. You would get a page with the parameter estimates and another page beginning with the results so that you can print all of that on a single piece of paper. You'll get a time stamp and date stamp on the printout. You can also download the results into an Excel file or a Word document, and those will both also have a time stamp and date stamp so that you'll know when you ran this particular analysis. Now, having gone through that first example, let's do what many of you will do, which is say, okay, I want to change something and see what happens to the results. Let's go back to scale of measurement and say, well, I'm going to recruit a heavier sample, and so the average BMI or the variance of the BMI might be larger with more variability. Let's just change that to 40. It's easy to do. As soon as I press continue, that is saved, and the 36 that I entered earlier is overwritten. I don't have to touch each of the other steps. I can go immediately to analysis with the same intervention effect estimate, calculate results, and I get the results now where the only thing that I've changed is that variance estimate. And with a greater variance, I need more groups per condition. And so now the answer that the software is giving me is 21 per condition instead of 19. And that's based on the simple change in the variance. If I go back to 36 and press continue again, now that's put the 36 back into the system. I could go to the interclass correlation tab and change it. Maybe I have evidence that it's not that bad. In fact, I can the interclass correlation based on my data may be closer to 0.025, so I'll enter that. Press continue. go to the analysis, calculate results, and now I get a different set of results that is different from the first one only by virtue of a smaller class correlation. And not surprisingly, I don't need as many groups per condition. You can do as many of these what-if exercises as you want. Let's take a look at what happens if I play with stratification or matching in the design. So I'll click yes instead of no. If I want to learn more about stratification and matching, there's a lot of information provided with references, so you're welcome to review that. Let's say I want to consider matching first. So I'm not going to use a priori or post hoc stratification, but yes, I want to consider matching. So I've said no here and yes here. Press continue, and I'm asked, what is the correlation between the matching or stratification factor and the outcome at the member level? I need a data-based estimate. Maybe I have one and it's 0.2. Enter the expected group level correlation between the stratification or matching factor and the outcome. If I don't have one, I'm going to leave that as 0 because that's conservative. The software has remembered my intervention effect of 1.5, and I just press calculate results, and now I'm getting results for a matched design where the correlation between the matching factor and the outcome is 0.2. I need 13 groups per condition or per study arm, and I have the same kind of tables down below. So as you can see, you can jump back and forth among all of these steps, make a change, go back to analysis and recalculate. It's very easy to do, and you'll get a new set of results based on that. So I'm going to click reset and start over. The software wants me to confirm that I really am serious. I want to do that. I'm going to back up to my slides because I want to show you another example. This one is an intervention designed to increase colon cancer screening. We plan to implement this particular intervention in community health clinics. We're going to verify up-to-date colorectal cancer screening via chart review in 50 participants per clinic at the end of the study. We expect a baseline rate of about 40% as being up-to-date on their screening. As before, we want 80% power, 5% type 1 error rate. We think that the intervention effect will be a relative increase of 30%, moving from 40 to 52. We base the variance on the average proportion. We expect it to be 40 in the control arm and move up to 52 in the intervention arm so that the average is 46%. We'll use mixed model analysis of covariance with regression adjustment for covariance and expect to explain a little bit of the variation at the individual level and the clinic level with our regression adjustment. We're not planning to match or stratify in the analysis. So now I go back to my sample size calculator. I'm going to look at the second example here, the numbers shown. I'm accepting the defaults for type 1 error rate and power. It is now a dichotomous outcome, no longer continuous. And the proportion that I'm expecting on average in the two arms is 46%. So I enter that as 0.46, not 0.46. If I put an extra decimal place in there, the software won't allow me to move on, so it catches that sort of mistake. Press continue. I'm still interested in an analysis of a simple difference. Press continue. The interclass correlation, it's whatever your data suggests it is, and in fact we have a published paper in Cancer Epidemiology Biomarkers Prevention suggesting that that ICC is 0.05, so we'll use that estimate. I said 50 participants per clinic. I said yes to regression adjustment, but no group, actually there will be group level covariance, so even though in my note over here I said no, we specified a reduction, so there I have to use at least 1 degree of freedom for the group level covariance. I said I would explain 20% at the member level and 10% at the group level. So I'll enter those and continue. No stratification or matching. The intervention effect, I said, was going to be a 12% increase in absolute terms, so 0.12. Calculate. And I'm provided with a summary of the parameters that I entered and given the result, which is 18. This tells me I would need 18 clinics at each study condition or study arm in order to have 80% power for an intervention effect of 12 absolute percentage points in colorectal cancer screening being up to date. So that's the end of my second example, and I'm going to stop at this point and see what questions we have from the audience. One question that's come from the audience is, do the methodology resources address new approaches like MOST and SMART? The methods that are presented here would address the final study that you're planning to do at the end of the MOST or SMART process. So those processes involve going through a series of studies initially to find out which components you're going to keep, and then you run the real trial at the end of the day to evaluate that. You could certainly use the sample size calculator here for that final trial. If you're going to use group randomization for earlier phases, you could use it for individual studies, but the resources are not set up to work through the entire MOST or SMART process with all of the pieces at one time, so it would be a piecemeal approach. The site asks for N per cluster. What if the sample size varies substantially by cluster? All right, that's a very good question. All of the, or most, I should say, of the publications on group and cluster randomized trials assume a balanced design so that I would have exactly the same number of observations in each cluster. I can promise you that that never happens in practice. I've been doing group randomized trials for 30-something years, and I never had a situation where I had the same number of people in each group. So long as the largest group isn't any larger than twice the size of the smallest group, you can generally ignore the variation. This is advice I got years ago from Leslie Kish. So if the largest group is no worse than twice the size of the smallest group, you can generally ignore it. But if the variation is greater, so that you have some groups that are 10 times the size of smaller groups, that's an issue. That is not addressed in this sample size calculator at present. We may be able to address that going forward, but it's not there now. Some of the references that we provided on the website for group randomized trials, and I'm going to go back to the website and go to the group randomized trials tab and come down to the sample size calculation. Some of these references address that specifically. For example, here's one, sample size adjustments for varying cluster size. So there are, and here's another one, relative efficiency of unequal cluster size. So there are references for the special methods that need to be used for that, but we haven't implemented them yet in the sample size calculator. Can this tool be used to compute the minimal detectable effect given a fixed available sample size? Yes, that's exactly what we did in the examples. So when we went to the sample size calculator, one of the steps, step 5, was entering the number of members or participants per group. And that's the number that you're starting with. You can always go back and change it, but the calculation is done on the number of groups per condition or arm that you need, given that many participants per group. So if you know that you're going to have 20 in each group, you just enter that. If you know you're going to have 50 in each group, you can enter that. If you know that you've got 1,000 across all of the groups that you're going to have, you'd have to divide that by the number of groups that you're thinking about to know what the number is. Another question. We need to determine the number of participants per group. How did you estimate the number? Well, I didn't actually do it. I let the software do it for me. Remember that we entered information on type 1 error rate and power on all of these other steps, and at the end specified an intervention effect and then pressed calculate, and the software gave us that answer. And the references that are provided will provide you with the formulas that are used in the spreadsheet. We may add those formulas later as additional information as part of the website, but they're not there now. Let's see if there are other questions that have come in. A question that is commonly asked is, why is NIH changing the forms and the procedures and instructions for clinical trial applications? This is a very planned effort by NIH to improve the quality of the research that we are supporting as an agency. We're very much aware that there have been problems in terms of reproducibility, studies that are done and can't be reproduced by others, and often that's because the methods are poor. So NIH is changing the general approach to clinical trials applications and is actually requiring a lot more information from applicants than we used to require. And we're requiring it to be submitted in a standard way so that everybody submits it in the same format, so that reviewers won't be confused by having it presented in one way in one application and another way in a different application. But the whole purpose of this effort is to improve the quality of the clinical trials that are supported by NIH. And the purpose of this particular website that I've been demoing today is to improve the quality of group randomized trials and individually randomized group treatment trials. Another question, will the review criteria be the same? No, the review criteria for clinical trials are going to be a little different than they have been in the past. Clinical trials were always evaluated using the same review criteria that every other kind of application was evaluated with. Going forward, there will be special review criteria, and you need to refer to the funding opportunity announcement that you're responding to see what those review criteria are. Remember that you can't just submit a clinical trial as an R01 to a parent FOA anymore, at least not after January 25th, 2018. You're going to have to submit in response to a specific FOA that explicitly allows clinical trials. And those FOAs will include the review criteria that your application will be judged against. A question that we've just received, can this tool be used to estimate power for 3-level mixed models with repeated measures on participants using the net difference feature? And the answer is yes. Let's walk through an example of that, and I'm just going to be making this up sort of as we go. Let's go back to the BMI example. We said we're going to use 5% type 1 error, 80% power, so we'll press continue. Scale of measurement continuous. Our variance was 36. And the software hasn't remembered these things, remember, because I pressed reset and set everything back to the defaults. This time I want to use an analysis of net difference. And now I get the chance to say, do I want a cohort design or do I want a cross-sectional design? In a group randomized trial, I can follow the same participants over time and therefore have repeat observations on both groups and members, or I can follow the same groups over time but measure an independent random sample of participants at each survey occasion, in which case it would be cross-sectional. So the information that I need to provide differs. If I want more information on these things, I just start pressing my expand for more info boxes and I get it. In this case, the question was aimed at a cohort design, so I'm going to pick that one. I'm presented with 2 questions that I haven't seen before. Enter the expected value of the correlation over time for the outcome at the level of the member or participant. We said that that was 0.7, so I'll just enter that number here. Enter the expected value of the correlation over time for the outcome at the level of the group or cluster. I don't know, so I'm going to leave it as 0. Continue. The ICC we said was 0.5, so I'll leave that at the default. We said we were going to have 25 participants per church, so I'll put that back in. And now regression adjustment for covariates. I'm still going to do it. I'm not going to have any group level covariates, but now I need to be thoughtful about what I put into as the proportion of the member level component of variance that I'm going to explain. I don't want to include the 49% that I think I'm going to get out of the overtime correlation because I'm not entering the baseline value as a covariate anymore. I'm including the baseline value in a repeated measures mixed model analysis of covariance. So since I'm not including the baseline measure, the amount of variance that I can explain at the member level is probably much less. I'm going to be conservative and suggest that it's 10%, and no benefit from adjusting for group level covariates because I haven't entered any. Press continue. Do I want to consider matching or stratification? I'll say no for now. The intervention effect is the same as it would have been before, 1.5 BMI units. Calculate results. And now I'm getting results for a repeated measures mixed model analysis of variance where I have observations nested within participants nested within churches. So it's the kind of three-level design that the question was asking about. I'm reminded that I've done the calculation for a cohort design with 0.7 correlation over time at the member level for the outcome variable. ICC is 0.05, 25 observations per church. I'm only explaining a little additional variation with covariates beyond what I can get out of the over time correlation. The intervention effect is the same. The results, a larger study. And this is often the case where I need a larger study to have enough power for the intervention effect that I need. In general, the analysis of covariance will be more powerful than the repeated measures analysis of variance because in the analysis of covariance, it's a net difference, a simple difference involving a comparison of two means or proportions or slopes. With the net difference, I'm comparing four means or slopes or proportions, and so I need a larger trial. And in this case, it turned out to be an appreciably larger trial. So that's an example of that kind of analysis. In terms of other questions, this webinar focused on changes to the application guide and review criteria and walk through this new website for group and cluster randomized trials and for individually randomized group treatment trials. And the question is, why did we focus on those methods? The reason is very straightforward. These methods are commonly used in biomedical research. We see them in animal studies. We see them in human trials. We see non-randomized versions. We see randomized versions. The analytic issues are the same, whether we randomize or not, and they're the same whether we're doing a study with animals or with humans. And because these designs are widely used and widely misused, we thought it was important to provide information on them on the website. As I reported earlier, only about half of the published studies, at least human studies, using group randomized trials, use exclusively appropriate methods for analysis and report appropriate methods for sample size calculation. We would like to see that much higher. The situation is even worse for individually randomized group treatment trials. We'd like to see that much higher. And so we've created this website to try to encourage investigators to become familiar with these issues and to propose studies that use appropriate methods. With the changes in the review criteria, we're also trying to bring this to the attention of reviewers so that they will pay attention to it when they're reviewing your applications. All of this is aimed at getting better applications, both submitted and coming out of the review process, so that we can have better research that's funded by NIH. Will other methods topics be added to the Resources for Researchers website? And the answer is yes, we hope that will happen. We focused a lot of energy on getting these first couple of topics up, and that's what's available right now. But we will turn our attention to additional topics. We haven't settled on what those are yet. If you would like to suggest topics, go to the feedback page, give us your name and email address, suggest topics that you'd like to see added to this website, and we would be happy to consider those suggestions. We very much encourage you to do that. Are there other questions that we've gotten online or through Twitter? Okay. Yes. Then I think, yes, we do, sorry. What ICC would be considered negligible enough to ignore if checked and found to be close to zero? Thank you very much for asking that question. I always enjoy having the chance to address that one. It's a very common question. I would say that there is no ICC that you should ignore. It's a very simple answer. Even very small interclass correlations can have very damaging effects to type 1 error rate if you ignore it in the analysis of the data from your trial. ICCs tend to be small. In most public health and medical research, they fall between 0.01 and 0.05, so they tend to be quite small. ICCs of 0.02, 0.03, very common. And you might be tempted to say, well, if it's that small, I can just ignore it. And it's just not true. It won't do much harm if you have very small groups with only, a dozen or 20 observations in each group. If the ICC is 0.001 and you ignore it, that might not do much harm. But as the ICC gets larger and you have more observations in each study condition or study arm, the impact of the interclass correlation is greater, and you can run the risk of having a type 1 error rate that's 20%, 25%, even 50% is not uncommon. None of us wants to have a type 1 error rate that's that high. Certainly NIH doesn't want it. And so my advice is always assume some level of positive interclass correlation based on data, not from your study, but based on published studies or pilot data. Part of the question was getting at what if I test the interclass correlation to see if it's different from 0, and the result comes back and says, no, it's not. Can I ignore it then? And the answer again is no. I don't recommend that. When you have a small interclass correlation, say 0.01, the standard error for that interclass correlation is not going to be well estimated. And the degrees of freedom to test whether that interclass correlation is different from 0 are usually going to be quite limited. So you're in exactly the situation where it is unlikely that the test is going to tell you that the ICC is different from 0. In other words, you're very likely to be told, no, it's not. But if you ignore it, you can have an inflated type 1 error rate. So it's a bad situation to test. I don't recommend ever doing it, and I always recommend taking account of the design that you're using in the analysis that you're going to perform. If the ICC is small, it won't have a horrible impact. If it's larger, you've taken it into account, but you should always plan for it, take it into account in your sample size calculations, and model the nested random effects in your data analysis. That brings us very close to the top of the hour, so I'm going to stop here and turn things back over to Jonathan.
Thank you, Dr. Murray, for all the useful information, and thank you to everyone who participated in today's webinar. On the Mind the Gap website, prevention.nih.gov/mindthegap, you will find several resources for this talk, including the slides, references, and a link to complete evaluation. Your feedback is very important to us as we plan future sessions. Thank you again.